Welcome to the online classes of animal biotechnology. Here I am going to discuss about the transfection of animal cell lines and their immortalization. I will discuss about gene knockout strategy in animal model, and finally I will discuss about animal cloning techniques and ethical issues. So first, transfection. So what is transfection and why and how we do transfect the cells? So transfection is the introduction of foreign DNA, RNA or protein molecule inside any eukaryotic cell. Transfection techniques serve as an analytic tool that facilitates the characterization of genetic function, protein synthesis, cell growth and development. Transfection assays not only enable the advancement of cellular research but also enhance the drug discovery strategies. So by the transfection we can know the fate of any drug molecule. So it is similar to the viral transduction and bacterial transformation. So here the types of transfection. So there are many methods of transfection to introduce foreign DNA inside any host cell. Like you can use calcium phosphate method. We can use viral methods like viral transduction methods. We can use chemical polypeptide, which is positively charged polypeptide, like lipofectamine. We can use this chemical for the transfection, and we can use the electroporation or electric pulse to introduce the foreign DNA inside the host cell. So here in this figure, I have shown the various transfected molecule like plasmid DNA is transfected inside the cell. Similarly, messenger RNA is transfected inside the cell as well as the siRNA or short interferic RNA is transfected inside the cell. Here, after entering inside the cell, the plasmid DNA can go inside the nucleus and it can transcribe the mRNA and form the desired protein. On the other hand, mRNA can directly synthesize the desired protein once it comes inside the host cell. And the siRNA is used to block the expression of the particular type of defective gene. So this is uh, type of negative regulation, means negatively it can inhibit the expression of a particular gene. So transfection is helpful in the expression of the desired protein as well as the inhibition of the defective gene. So how we can make the immortalized cell and why there is need to make immortalization? Because the cells go in senescence or cells may die after some time. So we have to, our main objective is to make immortalized cell. So how cell go to senescence? Because the chromosome of all the cells contain a telomeric region and the telomeres shortens after every cell division. So once the telomere starts shortening, the DNA start damage. And once DNA starts damaging, the tumor suppressive genes like P53 and RB genes prevent the cell proliferation by preventing the cell cycle and halt the cell division. And finally, cells cannot be divided in culture infinitely. So, how we make the immortalized cell? First, there are two strategies. First strategy, we can introduce the telomerase gene into the cell. Telomerase is an enzyme which can form the telomeric region in the chromosome. So we can, if you introduce the telomerase gene like STRT is the telomerase gene, this is one strategy. Another strategy is by the alteration of tumor suppressive genes. If you can alter or we can suppress the expression of P53 and RB genes, so in this way we can allow the cell for continuous cell cycle. So here you can see this is the telomerase or telomere which can extend the terminal part of the chromosome. And once this terminal part is remain intact, the immortalized cell has infinite lifespan and we can use them for long term project, like if you are working on a PhD thesis, from starting to end of your thesis you can work with the same cell without any deformity of the cell culture. So here I have shown that how it works. Suppose you have expressed the STRT gene or siRNA in the lentivirus or in plasmid. It can be transfected by any chemical like lipofectamine or other transfection method you can use to for the transfection of lentivirus or plasmid. Once it comes inside the host cell, here I have shown the one host cell. Uh, so here you can see the TRT gene is integrated in the host, and this uh, once this gene is integrated, the activation of telomerase takes place and telomere degradation stops. Once telomere degradation stops, on the other hand, the P53 and sbsiRNA can inhibit the expression of tumor suppressor genes. So you have to inhibit the expression of tumor suppressor genes because they can inhibit the cell cycle. So once you can inhibit the expression of P53 and siRNA, the cell cycle cannot be stopped. So cells multiply continuously. So here you can see the finally you got a immortalized cell which can divide continuously without losing their integrity or without losing their genomic constitution. So these cells be continually you can cultured in your laboratory and use for your research purpose. Now the gene knockout strategy or producing the transgenic animals. So what is gene knockout? As I already explained in my classrooms, that gene knockout means you have to excise a gene or a defective gene from the host by the homologous recombination method. So this is called gene knockout. Some other forms of gene disruption are also possible, like gene knockdown. In this case, you can reduce the expression of gene, or knock in. Like here, you can introduce a alternative gene inside the host cell. So for the gene knockout, three scientists got the Nobel Prize. Like here you can see their name, Mario Capecci, Sir Martin J. Evans, and Oliver Smith each got Nobel Prize for the creation of creation of knockout studies, and particularly they have worked in the mouse model and they created gene knockout mouse. So here the strategy of making knockout mouse or transgenic mouse. So there are two steps to making knockout mouse. The first is in vitro strategy. In vitro means you can do this thing in the outside of the living organism. So in this case, first step is embryonic stem cell culture. So you can isolate the embryonic cells from one mouse or from the blastocyst of mouse, and you can grow them in the laboratory condition. On the same time, you can make a construct which can uh, target a particular gene in the mouse embryonic cell line. So here you can see the construct or the cassette which can contain the neomycin transferase gene. Means once this gene is introduced in the embryonic stem cells, these cell can be able to grow in the neomycin media. So here you have make this construct, and you can transfect this construct into the embryonic stem cells. Once they can enter inside the embryonic stem cells, you can select them by the uh, growing them in the neomycin media. The only transfected cells will able to grow in the neomycin media. So here in the third step you can see the embryonic stem cell transfection. So once you have transfected them inside the embryonic stem cells, mm, the neomycin gene will be introduced inside their genome by the homologous recombination. Here you can see the how this neomycin resistance gene is inserted into the genome of the embryonic stem cell. So once this neomycin region is introduced in the look at this cell, it is introduced in the cell. So Further, if you will grow them in the neomycin media, only this cell will be differentiate or proliferate. So finally you will have the neomycin resistance embryonic stem cells. Now in the second step, from gene targeted embryonic stem cell to gene targeted mouse. Now you have in first step you have made the altered uh, embryonic cell or embryonic stem cell. Now you have to introduce this embryonic stem cell into the mouse. So for that, you target the embryonic stem cell and inject it into blastocyst of the another mouse, and where they mix to form a mosaic with the cell of their inner cell mass from which the embryo develops. So this now the embryo, the mosaic type means altered type embryo is developed, and this mosaic or altered embryo you can implant in the surrogate mouse or surrogate mother. So this surrogate mother gives birth to mosaic mouse, and this mosaic mouse means altered mouse. These altered mouse, once you mate this altered or mosaic mouse with the normal mouse, they produce both gene targeted and normal offspring. Like here, you can get finally the transgenic mouse where the both copy of the particular targeted gene have been replaced, and you can also get the normal mouse. So thus you can get the transgenic mouse here. Now the animal cloning. So as you know that the dolly was the first animal clone was created in the Scotland. So here I am explaining the strategy how the dolly sheep was created. So first the scientists took the uh, udder cell, the cells from the udder of a Finn Dorset sheep. This is a breed of a sheep. Finnish and Dorset is the breed of a sheep. From the udder of this sheep, they have took the donor cell, okay, and placed in the culture media. They grow it in the culture media, and the meanwhile they have also taken the egg from the another sheep, that is black face sheep. So they have taken the udder cell from one sheep and egg from the black face sheep, and they have removed the DNA from the egg. Here you can see the removal of DNA from the egg. And in the third step, both cells, the udder cell as well as the egg cell without the DNA have been fused by the electric pulse, and it was grown in the laboratory condition. After few days, like 5 or 6 days, embryos were developed, and these embryos were implanted into the another blackface sheep. This sheep now can act as a surrogate mother. And after the gestation period, the pregnant blackface sheep gives birth a baby, and that baby is very similar to the uh, donor sheep, means from where you have taken the udder cell. So this is the sheep which was developed very first in 1997 in the Scotland, and the name of this sheep was given Dolly sheep. Now there are some ethical issues also associated with the animal cloning, and these ethical issues are religious issues. I am not going to explain the religious issues, and there are some medical issues also. So medical issues means there are some it can cause some deformity in the clone. So here I have mentioned the various medical issues, like technically human gene grows older with age, and it is fair that the cloned individual would retain the age of the donor gene. So donor, it can retain the age of the donor gene forever. So that is also problematic, and the efficiency of this animal cloning is quite less. It is only 0.1 to 3% possibility because of following reason, like an nucleated egg and transferred nucleus may not be compatible. An egg with newly transferred nucleus may not begin to divide or develop properly, or implantation of the embryo into surrogate mother might fail, or pregnancy itself might fail. and some problem during later development also possible, like if pregnancy is possible, but that during the birth, the clone may be suffer from large offspring syndrome, means they have abnormally large organs. And third is there is possibility of abnormal gene expression pattern. So because you are fusing two cells from different individuals, so it is quite possible that the different gene have the change, different genes may change their expression pattern. And finally, the telomeric difference is also possible, that it is reported in the cows that the clone showed the signs of youth forever and seem to have the extended lifespan. Sometimes they have extended lifespan span of the clones, and sometimes they can less lifespan as the normal animals. So these are the medical issues of the animal cloning. So here I have summarized the pros and cons of the animal clones. Like pros, you can uh, eliminate the defective gene from any animal, or it is faster recovery from the traumatic injury. Infertility could also be eliminated by the cloning methods. Cloned body parts can serve as a backup system for humans. It can fight the genetic disease. It replicate animals for the research purpose, like knockout or gene clone. You can immediately use them in the laboratory for your research purpose, and it can produce people with the desirable trait. But there are some clones also of the animal clones. There are the possibility of faster aging or extended aging. There is reduced sense of individuality. It may reduce the overall value of human life. It weakens the diversity and ability for adaptation. It can produce undesirable traits also, and it invites malpractice in society. So there may be malpractices, means and scientists can also develop the human clone sometimes. So it is really dangerous. So that's all about the cloning. In next lecture, I will discuss about in vitro IVF or in vitro fertilization techniques. So till then, bye bye. Take care. Please stay at home, and thank you. Thank you very much.